Have you heard of quantum biology? Quantum biology asks whether quantum effects can influence processes inside living systems in ways that are biologically meaningful. In this recap, we are tracking what happened during the first half of the year. While this is not every finding, paper, or event from the year, we wanted to spotlight some of the moments that have happened in 2026 for quantum biology so far. Though it may not seem like it, quantum biology is a lot older than people might think. Its research spans a broad scientific landscape, from photosynthesis, enzyme catalysis, and magnetoreception to biological sensing, protein engineering, the effects of weak magnetic fields, and proposed applications in neuroscience, medicine, and more. Like I mentioned earlier, I'm not going to go into everything that happened this year, but let's talk about some of the research and papers that have come out so far. First, before we get into individual experiments, researchers spent time asking one question: what should actually count as quantum biology? In March, Gregory Scholes at Princeton and Graham Fleming at Berkeley published a perspective arguing that it's not enough to say quantum physics underlies biological chemistry. For Scholes and Fleming, the key question is whether a specific quantum process contributes to biological function in a way that experiments can distinguish from classical explanation. That gives the field an actual bar to clear. A separate review preprint mapped the landscape into three categories: quantum in biology, quantum for biology, and biology for quantum. Their assessment was cautiously optimistic. Some of the strongest current cases include tunneling in certain enzyme reactions and radical pair spin chemistry as a viable framework for magnetoreception. Many other proposed mechanisms remain scientifically interesting but unresolved. But that caution is not a weakness. It shows that the field is becoming more organized and explicit about where the evidence is the strongest, where it remains incomplete, and which experiments could move the science forward. One study investigated whether nuclear spin could affect the assembly of microtubules. Microtubules are structures built from a protein called tubulin. They help cells maintain their shape, divide, organize their internal components, and transport materials. And scientists compared different isotopes of magnesium. Magnesium-25 has nuclear spin, while magnesium-24 and magnesium-26 do not. The researchers found that changing the magnesium isotope altered how efficiently the tubulin assembled. The difference became even stronger when they introduced a weak magnetic field. A radical pair model was also able to reproduce important features of the experimental results. The findings show that magnesium nuclear spin can influence tubulin assembly in a purified experimental system and provide a new way to investigate how spin dynamics may affect cellular structures. Another paper proposed a new model for how migratory birds might sense Earth's magnetic field. Two leading explanations are usually discussed separately. One involves spin-dependent chemical reactions inside light-sensitive proteins called cryptochrome. The other involves tiny particles of a magnetic material called magnetite. Oxford chemist Peter Hoare proposed that the two mechanisms might actually work together. In the model, a magnetite particle amplifies Earth's magnetic field in its immediate surroundings. Nearby radical pairs inside a protein would then respond to the stronger local field, while still receiving information about the bird's direction relative to Earth's field. The model predicts that this hybrid system could produce a much stronger directional signal than radical pair chemistry alone. Researchers within our own ecosystem also reported new magnetic biology findings. Scientists studied an engineered fluorescent protein called MagLov2 inside living bacteria. They measured how the protein's brightness changed as the strength of the surrounding magnetic field increased. And the response was not a simple increase or decrease. Instead, it followed a non-monotonic pattern and became less responsive at higher field strengths. This behavior is consistent with predictions from the radical pair mechanism. Then a separate experiment examined what happens when bacteria are grown in an environment with almost no magnetic field. Scientists from the same Quantum Biology Institute grew E. coli inside a hypomagnetic chamber that reduced the surrounding field to about 19 nanotesla. For comparison, Earth's magnetic field is usually measured in the tens of microtesla, making the experimental field thousands of times weaker. Under the hypomagnetic condition, the bacteria remained in the lag phase, the period before rapid growth begins, about 50% longer than bacteria grown under normal Earth-strength magnetic conditions. Once experimental growth began, however, the two groups grew at essentially the same rate. Under these experimental conditions, the magnetic environment appeared to influence the transition into active growth more clearly than the subsequent exponential growth rate. Researchers also tested a radical pair model in planarian flatworms, organisms known for their remarkable ability to regrow missing tissue. Rashab and collaborators predicted that levels of superoxide, a reactive oxygen species involved in cellular signaling, would rise both near zero magnetic field and at fields above approximately 500 microtesla. They then tested that prediction experimentally and observed the same general pattern. The exact radical pair involved has not yet been identified, and the superoxide changes did not directly translate into simply more or less tissue growth. The results support the idea that radical pair spin dynamics may contribute to magnetic field effects on superoxide during regeneration. More broadly, this study shows how quantum biology can move forward through a clear sequence, propose a mechanism, make a testable prediction, and compare it directly with the experiment. Additionally, Abrahams and colleagues published a paper in Nature describing engineered fluorescent proteins, like mentioned earlier, called maglov, that can act as quantum sensors inside living bacterial cells. The researchers optically detected magnetic resonance, or ODMR, from these proteins at room temperature. By applying static and radio frequency magnetic fields and measuring changes in fluorescence, they were able to detect the protein's spin-dependent response with enough signal to measure individual cells. They also demonstrated several possible applications, including distinguishing protein variants, detecting changes in the protein's local environment, and locating fluorescent signals using magnetic field gradients. Finally, a review examined where quantum biology could intersect with medicine. The review connects proposed quantum biological mechanisms to specific questions involving metabolism, redox signaling, cancer, and neurodegeneration, helping outline directions for future biomedical research. Taken together, these papers showed just how broad the field is becoming. But research was only one part of the story. Across the first half of 2026, the researchers, communities, institutions looking into these ideas also became more connected. The year began with Quantum Biology in Nigeria, an educational initiative out of our own DAO that brought students, researchers, and scientists together around quantum biology. Organized by community members with a university there, the event demonstrated that participation in frontier science does not need to remain concentrated within a small number of established institutions. Building a global field also means creating credible entry points for communities that have historically had less access to specialized scientific networks. The Biophysical Society also hosted a dedicated event on quantum biology and biophysicists, co-organized in conjunction with our DAO as well. The program covered electronically excited states, magnetic field sensitivity, the relationship between biophysics and quantum biology, and the future of scientific funding. This is a sign that quantum biology is increasingly entering conversations with neighboring scientific fields. And then across the spring, the Guy Foundation organized their lecture series on magnetic fields and biology. The sessions explored the radical pair mechanism, hypomagnetic environments, magnetic sensitivity in plants, animals, and humans, and possible implications for medicine and space travel. Then in April, things picked up fast. Northwell Health hosted the inaugural Quantum Biology Forum at United Therapeutics in Silver Spring, Maryland. Only days later, scientists gathered at the Niels Bohr Institute in Copenhagen for Frontiers and Challenges in Quantum Biology. The location has a particular historical meaning. In 1932, Niels Bohr developed his influential Light and Life lecture in Copenhagen, where he considered how ideas from quantum physics might relate to living systems, if that sounds familiar. Nearly a century later, researchers returned to this city to discuss even more questions in the field. Then moving into summer, people gathered in Budapest for the 15th annual Quantum Effects in Biological Systems workshop, better known as QEBS. QEBS is one of the field's longest-running dedicated scientific meetings. The program covered quantum effects in biology, quantum technologies for studying biology, and biological systems that could support quantum technology. Our own DAO was even able to participate, presenting our work on decentralized science funding models and community-driven research. Only days later, the Quantum Biology, Cognition, and Medicine workshop took place in Sweden. And the following week, the University of Surrey hosted the Sustainable Quantum Innovation Workshop. Its focus was not only what quantum effects might occur in biology, but also on whether nature's efficient biological processes could inspire more sustainable experiments or technologies. Quantum biology also appeared in gatherings focused on medicine, cognition, technology, and more. This expansion into neighboring disciplines is an important signal of growing scientific interest and is creating new opportunities in collaboration, experimentation, and in the exchange of ideas. Taken together, the first half of 2026 showed the field becoming more organized, more connected, and better equipped to test its central questions. Researchers worked to define stronger standards of evidence, tested proposed mechanisms in living systems, developed new experimental tools, and brought quantum biology into wider conversations. Those conversations are continuing across conferences, laboratories, and even online communities, helping more people discover the field and contribute to its growth. And while progress is happening across many areas, the field still may be approaching a big breakthrough that brings these ideas into a sharper focus. Looking ahead, we hope to see more independent replication, stronger experimental evidence, new collaborations, and clearer answers about which quantum mechanisms actually influence living systems. Subscribe for more updates from across the field and join our Discord if you want to be part of the longer conversation of accelerating the field of quantum biology. The first half of the year continued to build momentum, and let's see where the rest of 2026 and beyond takes us.